Okay, so on day one, we had the bromeliad gene and the splicing with that, the different parts. There's a limited area with the gene, so because of that variation in the eukaryotes with alternate splicing, and then like the overview of the lab, then we get to the bacteria, and then they'll hopefully code the protein after the plasmid is added. And then there's also an operon involved with that gene expression. And then next we're gonna isolate the hopefully a mass amount of glow protein, and then the dogma is the DNA, RNA to protein. And remember, there's a promoter region on the DNA, and then there's a terminator region to stop the RNA, and then gets coded into the protein. Day two is there is a leaf, and then we put that leaf in the solution, and we remove the nucleus to get to the DNA with soap. Arabinose was it was the allosteric property that changed the repressor protein on that operon. And so, is it an operon for a gene? Yes, it is. It's for the creation of enzymes to digest that sugar. Hind 3 was the restriction enzyme, so it looks for a specific line of bases, and it'll cut the gene right there, and then the bases will attract each other. Remember, when you're splicing all those loose ends, the ligase will combine them. Day 3, this is the transformation of E. coli, and then remember, it's a bacteria, it's a model organism. You can make a bunch of protein that way. You cool and heat to add the plasmid, which is like an extra part of the gene code that's in the bacteria, to the circular chromosome to insert it into that. And then there's the two plates, the petri dishes, and then there's plus arabinose and none in the other. And then both of them had colonies, and then hopefully they're transformed. There's no indication yet, but then later on there will be that we hopefully transformed all of them, and then of course the expression. Day 4 was we found out that the plus arabinose glows and the other didn't. So that must mean in this one, because of the presence of the arabinose, there's a glow gene which made the protein through the dogma. Then we put them in culture tubes with the broth, the nutrient broth, and then there's one for the specifically the ones that were glowing, and then one that was for the ones that weren't. Uh, would the glow gene be in everything? Again, we assume the plasmids had it, and then they transform them. So yes, but there isn't the indicator in the other one that it was transformed because there wasn't the expression of the glow. Um, what caused it to glow? We could hypothesize that the arabinose had something to do with an operon that expressed the protein. And once again, on some of the things, you can't tell if it was transformed. On day 5, we prepared to remove the bacteria. So they both glowed in the broth in those culture tubes. So that must mean arabinose was in that broth. So we move them to the microtube, and the centrifuge, this thing that spins it around the centrifugal force, it will separate it by weight. But not chemically. We want to separate the protein by chemical means later on to isolate it. So in the supernatant, remember that's like not the pellet. The pellet's the hard stuff at the bottom. The supernatant is the aragonose plus the nutrient broth, and the bacteria in the pellet is there, but the cell wall wasn't destroyed, so you'd find the glow protein there if there was any. We need to redissolve the pellet with a buffer before doing the next step, and then we also add lysozyme, which is an enzyme that breaks the cell wall, again taking away one of those components that we don't need. We need to get closer to the protein, which is in the bacteria, hopefully in the bacteria. And then now we add water, and with the freezing and expansion of the cytoplasm in the cell, again, it'll just release the inner contents and the proteins in the bacteria. Okay, so that's day 5. Day 6 was separating cell parts from protein. Again, the centrifuge, because this is like a physical separation, not a chemical one. So afterwards, the supernatant and the pellet again. A supernatant has all the proteins and the glow protein, hopefully. The debris will be at the pellet because it's more heavier. And we can't inject it yet because it might have some bad side effects. It's from the lab into a living organism through testing. Can't do that yet because there's other proteins in there that might be harmful. Our next step actually would be the AA chart, but we could combine them because we kind of did them late. Anyways, chromatography was separating by mass and rate via a chemical process using that buffer zone here with the beads that are hydrophobic. So there's going to be some chemical relations, hydrophilic and hydrophobic, referring to like the charges and the attraction between the molecules involved. I'm sorry, I have a sore on my tongue. Okay, so that's the chromatography. That's like a diagram we had to draw, and then day 7 was actually the separation. So on the AA chart, so the glow gene, and then we took the RNA, and then we looked up the amino acids. Most of them were hydrophobic. So remember, hydrophobic things bind to hydrophobic things, and hydrophilic things, bind to hydrophilic things. These don't like each other. It's like a magnet. They'll push away. Okay, so salt solutions. There are 3. Remember, there are 3 things that we put through this chromatography tube and the Weiss tube at the bottom. So the first one's the binding. So it was high saline solution. The hydrophilic amino acid proteins went out into the waste. You'll see here the protein. Remember, when it's put in a hydrophobic or a high solution, the hydrophobic glow protein, it folds the protein up, and it would want to bind to the beads. And the second time, the binding is the washer solution was a medium salinity, and the ones that came out of this into the waste zone here were the partially hydrophobic amino acid proteins. The mostly hydrophobic proteins were still around the beads, but they're still loose. They're loose now a little bit. And then finally, the TE wash was a low saline or none saline at all. The glow went out. And so, yeah, the glow went out. It was separated from, this is the most hydrophobic protein. So then it went out when we added this. And that's about the lab. I think there's a lot of other things that are covered in the other papers. Now I'll just take another picture and send it. Okay, bye.